Alrighty, so we're going to be talking about gene regulation in eukaryotes. There it is. Billy, can you get the lights over there real quick? I appreciate that, so you all can see it a little bit better. Okay, so looking at this diagram real quickly, this is your DNA right here. Okay, if this is DNA and this is RNA polymerase, what does the yellow represent? What do you think that yellow represents? The RNA polymerase. It's a gene potentially, but what does our RNA polymerase always bind to? The promoter. Okay, so this is your promoter. And yes, even though eukaryotes do not have operons, they have some of the same elements of operons. They have a gene that's going to be transcribed. They have a promoter region where RNA polymerase is going to bind to, but that's about it. Then there's a lot of other differences between it. Okay, so we're going to talk about those a little bit. All right, so this is chapter 18 in your textbook, Regulation of Gene Expression in Eukaryotes. And there's basically two levels of control that we see in eukaryotes. Short term control, that's kind of like turning a gene on to make an enzyme to break down, lactose that's all of a sudden in the system that's not there. Okay, we also do that. We create those genes. It's just regulation of day-to-day activities in a cell or a group of cells, i.e., a tissue, in order to maintain the function of that organism or that particular cell. Long-term control is dealing more with the formation of the embryo, of the fetus, and developmental procedures. Hey, we're going to turn this gene on now because we want a head right there. Okay, we're going to turn this gene on because we want two arms at this point, and then two legs and a torso. So those particular genes get turned on at a very particular point in your life cycle, and then they never get turned on again. Okay, so that's more of that long-term regulation and development. Prokaryotes don't really have that, right? Because they're constantly dividing and they don't really have this long-term lifestyle or anything of that nature. So, as we all know, all of the cells in our body basically have the same DNA. Yes, there might be a few mutations here and there that make some cells different, okay, but in all practical purposes, they are genetically identical to each other, okay? Only 30 to 40% of all the genes in a particular type of cell, maybe it's a skin cell, or maybe it's an epithelial lining of your stomach or your digestive tract. Only 30 to 40% of those genes are being expressed at any point during that cell's life cycle. The rest of the genes are turned off. They're not being expressed in that particular cell because you don't want to have a fingernail gene being turned on in your eyeball, right? That would not be very comfortable. Okay, so those genes are permanently turned off. If we're talking about highly specialized cells, such as a nerve cell or maybe a muscle cell, which are really specialized, even a smaller amount of genes, maybe only 10% of the genes are actually turned on in those particular, or get turned on in those particular cells. So the question is, how are those genes turned off long term, and then how are they turned on short term? Those are the two questions that we want to look at today. Okay? And again, it all comes down to what we call differential gene expression. Some cells are going to express certain genes at some point in their life cycle, and they're never going to express the other ones. Okay? So you got a lot of differences, a lot of words up here and things like that. But we do know, of course, in prokaryotes, we have these operon systems where we have a regulatory gene, we have a an operator, we have a promoter, then we have the multiple genes usually can make up that operon. As I said earlier, eukaryotes have some of the elements there, mostly the promoter region and a single gene, but they don't have all the other stuff. Okay? Eukaryotes, however, do have some other things. All right, like I said up here, eukaryotic genes also are regulated in units of protein coding sequences. So we have the promoter, we have the gene, and we have some regulatory molecules that can help to play a role in turning that gene on and off that are generally grouped relatively closely together. And genes that are related, let's say we have a series of genes to make tryptophan. They might be they might be on the same chromosome. They might be kind of close to each other. But unlike in an operon in a prokaryotic cell, we do not have multiple genes being transcribed and translated with the start of a single promoter. No, that doesn't happen. Okay? It's much more complex, again, because of the possession of a nucleus. So this gives eukaryotic control at many, many different steps because of transcription and translation occur in different places at different times. Each one of those places and time differences can be a point of control of the final product. Okay, so we have multiple points of control. A prokaryote, the gene's turned on or it's turned off. I should say genes because multiple genes get turned on in an operon system. We either turn it on or we turn it off, and that's it. Okay? In eukaryotes, we can adjust lots of different things. And again, the two categories of gene regulation, the short term and the long term. Short term, daily function of that particular cell or group of cells or that organism, and long term, again, generally playing a role in development and differentiation. What do I mean by differentiation? Starting off with like a stem cell, and then that stem cell giving certain signals to different cells, and those cells start to specialize. They become a blood cell, they become a bone cell, they become a liver cell, something of that nature. And once they go down that road of becoming one type of cell, they don't go back. Okay, they don't go backwards. They will always that lineage will always be that type of cell. Okay, so eukaryotic gene expression, six different levels, at least six different levels, and we're going to go over these levels today. First level at transcription. Do we turn a gene on or do we turn it off? The second level, RNA processing. Okay, how do we process that RNA? And lots of different things can be done. Because what happens in RNA processing? We're taking out the introns. We're putting those exons together, not always in the same way, because we can put them together in different ways to make a different product. If you're putting a cap on there, what's the cap do? It helps direct where that messenger RNA is going to get translated. Okay, whether it's on the RER or whether it's on a free-floating ribosome. Okay, that's going to affect the outcome. How long is that poly A tail? That's going to determine how long that particular messenger RNA is active. So lots of things can happen here. Okay, RNA transport, does it leave the nucleus? If it does leave the nucleus, where's it going? And again, that's where that 5' cap helps to play a role, going to one of those two areas. Messenger RNA translation. So once the messenger RNA is out here, okay, exactly, again, which ribosome does it go to, free floating or a rough endoplasmic reticulum? How long is that translation process going to occur, and so on and so on? How long messenger RNA degradation? Again, part of that is determined up here in the RNA processing when that tail is put on. How long is that tail? How long will that messenger RNA be there making its protein products? And the last one is protein modification, which happens as it goes through the endoplasmic reticulum or through the Golgi apparatus. And finally, how long does that protein last in the cell, okay, before it starts being broken down and degraded? So all of these are various steps in the outcome of those proteins. Versus in prokaryote, turn the operon on, make the protein, boom, that's it. Okay? But here we have multiple steps where we can control it. Okay? So lots of different things going on. We all know that the cell, its DNA is in the form of chromatin when it is doing its job, its active job. The only time we see the chromosome is during mitosis or meiosis. And again, that's as we talked earlier, that's when basically all cell functions are shut down because the DNA, the genes in the DNA, are inaccessible because they're all tightly wound up into a chromosome. Okay? In order for the transcription factors and the polymerases and things of that nature to land on a promoter region and actually transcribe that gene so it can ultimately be translated, the DNA has to be kind of opened up, has to be in that chromatin form. Okay? So one of the other differences too that we see in eukaryotes versus prokaryotes is eukaryotes have these specialized proteins, these histone proteins that the DNA is wrapped up around. And again, if you've ever seen like a big spool of wire on the back of a truck when they're doing line work, or if you sew and you got a spool of thread, think of that spool as being the histone, the protein, and the thread or the wire wrapped around that spool as being the DNA. Okay, that helps to package the 6+ feet, 2+ meters of DNA that you have in each one of your cells. Okay, so think about that. Let's say you got 2 meters of DNA in each one of your cells. The last estimate I saw was you have 30 trillion cells. Okay, that's 60 trillion kilometers or 60 trillion meters. Okay, 60 trillion meters. That's a long way. That's like out to Pluto and back. Okay, it's far. All right, it's far. So, anyways, you get a lot of DNA in your body. That's all I'm saying. You got a lot of it. All right. So, in order for the DNA, the gene to be transcribed, the DNA has to be kind of loosely, can't be compacted up into the chromosome form. So normally, what we see, we rarely ever see the DNA just opened up like a helix. Generally, what you're going to see are these little beads on a string, where the beads are the histones. Okay, and here you can see a picture of the little dark spots are the histones, and they're all joined together by DNA. This DNA is in its active form. It is ready to be transcribed. And this is what, when a gene is getting ready to transcribe, that small section of the DNA will look like this. Okay? The rest of the DNA is going to be in this chromatin form where it's more compact. Okay, not all the genes are easily accessible and stuff like that, but it's still it's not hard to open this up a little bit and the gene can transcribe that. As the gene moves into mitosis, those chromatin fibers start to condense in the chromosomes, and from here onwards, those genes get really hard to get access to. So a working cell, most of the DNA is going to be in one of these two forms, nucleosomes or in chromatin fibers. The rest of the stuff, the DNA is turned off. So here's a cell during interphase, okay? And the two different types of chromatin that we didn't really talk about it. I just called it chromatin. But there are two major types of chromatin in a working cell, i.e., a cell in interphase. We got the euchromatin, which is all of this light gray speckled stuff, and then the heterochromatin. That's these big dark blobs. The heterochromatin is DNA that is basically permanently shut off in this cell. Okay, any of the genes that are found in this heterochromatin region will not be turned on ever. Okay, it is shut down. Okay, so this is a cell after it has become specialized. Okay, it's never going to turn those genes on that are in that heterochromatin form. But the stuff that's in that euchromatin form, those genes can be turned on. And what's interesting, when this cell prepares to divide and replicate, the two daughter cells will have the exact same regions that are in the heterochromatin form. So it passes this on. Okay, it passes this DNA inactivation onto future cells. Okay, so that's what cell specialization is basically, is that major portions of the DNA in any particular cell that has become specialized are permanently turned off. Okay, what kind of cell do you think would have all the DNA open, where all of it's ready to be transcribed? Very special type of cell. Might have heard of them. Embryonic stem cells. Okay, and you've probably heard of embryonic stem cells. Those cells are what we call totipotent. I think I mentioned that term before. And a totipotent cell is a cell that can make any type of cell, plus it can make the cells that will become the placenta in the mother's uterus. Okay, that is a embryonic stem cell. You also have cells that are what we call pluripotent, which can make any type of cell, but they can't make the placental cells. So in humans and most other mammals, if I believe correctly, if I remember correctly, I should say, our cells are only totipotent to about the 8 cell stage, maybe 16 cell stage. I don't think if you go beyond that, it doesn't. They're no longer totipotent. They become special. They start to become specialized. So what do I mean? You start off with the fertilized embryo. It splits into 2 cells. That splits into 4 cells, and it splits into 8 cells, and then it goes into 16 cells. However you do that, okay? The embryo, the size of that doesn't get any bigger. It's just that the cells are getting smaller and smaller and smaller. So up to about the 16 cell stage, when you have 16 little cells all in there, okay, you can go in, and we do this with animal husbandry, is that we can take this 16 cell ball of cells out, separate them out, and implant each one of these individual cells into a donor egg, and then implant that into a female, into a cow. Okay, female cow is kind of redundant. Okay, all cows are female. Alrighty. So you can do that. So that way, what they do is that you'll end up now with 16 identical clones, and that's how they do a lot of animal husbandry. Okay? So again, with humans, if you twins, you start off with a single fertilized egg that splits into 2 eggs actually, and of course they're genetically identical. So you end up with your identical twins and things. Any question on euchromatin versus heterochromatin? Okay, do you know those? So here's our DNA control stages in protein synthesis. And again, we're going to start off with in the nucleus. Okay, in the nucleus and DNA structure. So what causes heterochromatin? Methylation. All right, methylation. What's methylation? Remember a methyl group we should remember is CH3. That's a methyl group. And you should remember methyl groups, are these polar or non-polar? They're non-polar. Okay, because remember carbon-hydrogen bonds are non-polar bonds. Carbon-oxygen bonds are polar, are polar. So this is non-polar. Is non-polar hydro or hydrophobic or hydrophilic? It's hydrophobic. Okay? And of course, polar bonds are going to be hydrophilic. All right? So, DNA methylation. Here is your chromatin. Here's your histones. Your histones have little, I'll show you another picture pretty soon, but basically what happens is that methyl groups get attached to these histone tails right here. And so here's an unmethylated piece of chromatin. Transcription is possible because it's nice and spread out. You add methyl groups, which are these little red dots right here, and that causes the nucleosomes to get really close together, and now it's going to prevent DNA transcription. How do they do that? Well, this is hydrophobic. What's the environment inside a cell? Water, right? It's mostly an aqueous environment. So these things want to get away from the water, so they cluster together, causing this to bunch up. Okay, causes it to bunch up. Take the methyl groups off, and they kind of open up. But there's another way we can open them up also. Okay. Um, the other way we can open them up is by Here's your histone proteins, and the histone proteins actually have these little tails that come out of them, and their amino acid change is all they are, and methyl groups attached to these. They're amino. These are the R groups coming off. Methyl groups can attach to them. They can make them hydrophobic. But then what can also happen is that you can add acetyl groups. Remember in cell respiration, in the process of going from pyruvate into the mitochondria, we had to take off a carbon and we created this molecule called acetyl CoA. Okay, acetyl groups are polar. All right, they like water. They're hydrophilic, and when you add acetyl groups, multiple acetyl groups to these histone tails, that causes the DNA to spread open, become open. Okay, so we can permanently open up DNA, or we can permanently shut it down by adding methyl groups, or permanently open it by adding acetyl groups. These things, are they changing the DNA, underlying DNA sequence of nucleotides? No, they're not. These are molecules on the outside of the DNA. And you've probably heard this term before. These are what we call epigenetic modifications. Okay? Epi, if you remember your prefixes from ninth grade, means on top of. Okay, so an epiphyte is a plant growing on another plant, like a fern growing up in a tree or Spanish moss on a tree. Those are all epiphytes. Epigenetics are modifications to the underlying genetic material, DNA. Okay, and in this case, there's basically two types of molecules: acetyl groups open it up and promote transcription, or methyl groups, which close it down and basically prevent transcription. What's interesting, yes, ma'am.
So does acetylation go with euchromatin?
What's that?
Does acetylation go with euchromatin like methylation does with heterochromatin?
Yes. Okay, it plays a role in chromatin. All right, and what's interesting with these is that we don't evolve. We Our DNA was set when sperm met egg. Our DNA does not change. Okay, unless we get zapped with high doses of radiation or something like that, and that would have to happen to our gonads so we can pass it on to future generations. But mostly our DNA does not change. However, what we can do through our actions, good or bad, I'm going to smoke a bunch of cigarettes, drink a whole bunch of alcohol, sit around and do nothing. Okay, you can add you can add epigenetic factors to your DNA that you can now pass on to your offspring. Okay, and the same thing is obvious. You can add good epigenetic factors that you can also pass on to your offspring through lifestyle choices, or sometimes not lifestyle. They first discovered this in a small Dutch town that was surrounded by the Nazis during World War II. And it turns out that the children born from those people that were trapped in those villages had a much higher rate of obesity than ones that were in maybe a neighboring village that were not surrounded by the Nazis. What the heck was going? What did the Nazis have to do with it? They were trying to starve the people out. They were because there was resistance cells. Remember I talked about Jacques Monnet the other day being a French resistance fighter. Well, during World War II, there was all these like resistance fighters in all these countries, and the Nazis were trying to basically starve them out. So the people that were living in these particular villages were basically dying of starvation, and that caused epigenetic changes to their DNA, okay, that were later discovered like in the 1990s by researchers wondering why were these clusters of people, with obesity, and in the nearby town, another nearby town, there weren't any of these people, people that had these clusters of obesity and things. So pretty interesting stuff that the discovery of these things. And so again, this is how we can, again, affect our offspring, affect our grandchildren, if you decide to go that route, okay, in the future. So we cannot change our underlying DNA, but we can change these epigenetic factors that may cause good or bad things later on in the future, in yourself and also in future generations. So pretty cool stuff there that's going on. And again, that's all relatively new. Within the last 20 to 25 years, this work has all been figured out. All right, so we can either have the DNA in a form that can be transcribed, or we can have it, which would be the euchromatin, or we can have it so it can't be transcribed, which would be the heterochromatin. So now, once it can be transcribed, okay, what are some of the changes we can do? What are some of the modifications that can happen? Well, just like we saw in prokaryotes, there is a promoter region at the start of a gene. And I'm saying a gene and not multiple genes because that's the way eukaryotes roll. Okay? In that promoter sequence, there's also something we call the Tata box. Okay? Tatas. All right? And it's basically just a bunch of T's and A's. Okay? That's why it's called Tata boxes. These this is so we know the promoter site is where the RNA polymerase binds, but the Tata box is where certain types of transcription factors, which are also proteins, will bind to that. And if we don't have a transcription factor there, RNA polymerase will not bind. Okay. Now I want you to look at this bottom picture. We see one, two, three, four, five, six different transcription factors. This is a hallmark of eukaryotic DNA transcription, is that there's actually a factory, if you will, of different proteins coming together to make to start the transcription process. Versus what we saw in prokaryotes, where it might be one or two different proteins, but here there's a whole bunch of them. And again, this is only part of it. This is the all right. There are also other series of proteins here that we call enhancers. I want you to notice up here. Here's the gene, here's the promoter. These enhancer regions can be pretty far away from that particular gene. Now, if we have several genes that might be playing a role in a certain biochemical pathway, all those genes would have to be transcribed pretty much simultaneously in order for that pathway to work. And so many of those genes have the same different types of activators and enhancers. So when those activators and enhancers are transcribed and translated, it will turn on all of those genes at the same time. And those genes might even be on different chromosomes, but the same types of enhancers will turn those particular genes on. So what do we got here? These are all these transcription factors and proteins that all have to come together, okay, to bind to this. But this RNA polymerase will not move until these molecules come over and basically bind to these mediator proteins. A lot of stuff going on here, guys. Okay, so that has to happen. And this is all happening at the point of transcription. That's the only place we've gotten so far, these enhancer molecules. This diagram, a couple of these diagrams are in your book, but I want you to look at why, this is basically telling you why eyeball genes are not turned on in your liver and why liver genes are not turned on in your eyeballs. It has to do with these activators. Okay? Not all cells are going to have the same activators in their cytoplasm. We know all cells have the same DNA. Okay? But we do also know that some of that DNA some of it's in euchromatin form, some of it's in heterochromatin form. But there's also these activators. Okay? If a cell does not have the activators necessary to turn on the eyeball gene, this is for making your lens, that gene will never get turned on. Okay, that gene will never get turned on. So let's kind of just walk through this diagram. This is your albumin gene, and it has certain enhancers that it needs to have. It's got yellow, gray, and red. This is your crystalline gene, which makes your lens. It has orange, gray, and pink. They both have gray. Okay, they can have the same kind. but every gene has a unique combination of enhancers. Okay, so unless there's multiple genes that are necessary for a certain biochemical pathway, there might be several genes might have the same enhancer combination, but all those genes would have to be active at the same time. So there's that there. So again, if you'll never turn on the crystalline gene in a liver cell because will not have the activators that are necessary, which are going to be the pink and the orange. Here we got pink and orange. This is in the eyeball. Those are available to turn on that particular gene, but they're not available in the liver cell. So again, you have to have all the right ingredients in a particular cell in order for those genes to be turned on. Okay. Messenger RNA degradation. So we made the RNA, it's made it out into the cytoplasm. Okay. So a couple of things can happen here is that one of the things is that messenger RNA there can be other molecules being made that are going to break down that messenger RNA. We call these small interfering RNAs or sometimes microRNAs. Um, we use these a lot in genetic technology, but don't really know the details of this. Just know that messenger RNA degradation can happen relatively quickly. Okay, and there's two ways that can happen: having a short poly A tail or through these microRNAs. I'm not going to really get into a lot of detail there. Okay, we've already talked about RNA processing. This is another stage, and this is also happening after the pre-messenger RNA has been made. We can make a variety of different proteins through alternate splicing and dicing. Remember, we take the introns out, we splice them together, but we can also do it in different orders. The different orders will give us a different protein product. Remember the 5' cap, that's going to tell us where this messenger RNA goes. Does it go to a free-floating ribosome? Does it go to the rough endoplasmic reticulum? Poly A tail, how long that messenger RNA is available. Okay? Once the messenger RNA leaves the nucleus, it's going to go into the cytoplasm where it will bind with the ribosome. Okay? And that ribosome, of course, is going to cause translation to occur. Now there's also those small interfering RNAs can actually bind with the ribosome and prevent translation from happening. So different things can happen. The other thing, if that protein is going to stay in the cell, we have these other proteins we call chaperones. Think of it as I don't want to do that, but we'll use this one here. Okay? So here's a chaperone. It's a protein. All right? Here comes the protein. It's just been made. It's a long strand of polypeptides. Well, if it's just in the environment of the cell, it might fold up into a couple of different ways. It's in this watery environment, but it might not be the correct way. Maybe this protein, in order to be folded, has to have a special environment around it. Maybe an environment that is more hydrophobic, okay, more non-polar. And so these chaperone proteins provide these special environments, and there's a whole bunch of different ones. And basically, the polypeptide goes into that, and in that special environment, it's going to get folded into the correct shape that it needs to be. Okay, and then when it comes out, boom, it's going to be in the correct shape. And again, got kind of interesting names. What do chaperones do? They make sure you're doing the right thing. Okay, chaperones are making sure proteins fold in the right order and in the right shape. Because if it's not folding in the right shape, it's not going to do its job. And again, we already talked about if that protein is going to leave the cell, then it has to get translated in the endoplasmic reticulum. Um, and again, the last place where we can see control in eukaryotes is how long that protein will be available in the cytoplasm. Okay, and there are a bunch of large protein complexes called proteasomes that will break down proteins. Okay, and they have special enzymes in them called proteases. which will break down those proteins. So if the protein is only needed for a very short period of time, boom, it'll get first of all it'll get tagged with another molecule. Don't need to worry about the names of that. It'll be tagged, and once it's tagged, it'll be destroyed and then recycled from back in that cell. So kind of going through this whole thing. Changes in the structure and function of the chromatin can turn on or turn off genes permanently in a particular cell. Okay, transcription. When that gene gets turned on, we need to have lots of different specialized factors in eukaryotic cells for that to happen. Okay, RNA processing. Again, things that do not happen in prokaryotic cells. How do we splice together those exons? How long do a poly A tail do we put on? What type of 5' cap, which will determine where it goes to get translated? How long does it last once it leaves? How long does it last in the cytoplasm? Where does it get translated? And finally, how long is that protein available? Okay, so there's one diagram in your textbook. I think I marked it on the chapter 18. This one right here, basically, that's in your textbook in chapter 18. Give you a nice little breakdown of all the various points where eukaryotic gene control can happen or protein control can happen. So that's all of that stuff right there for eukaryotic gene control. A little bit more complicated than prokaryotic gene control. Now, prokaryotes with those operons can sometimes get a little confusing, but just remember the lac operon and the trip operon as examples of of inducible and repressible operons. Okay, look through those pogles again, and that should get you right there. All right, so I'm going to stop for today, and what I want you guys to do, I would use some of this